Cool. So today we'll talk about this paper that deals with chronic stress and Alzheimer's disease. And I didn't really realize until I started reading the paper that we had discussed already a paper from this group. So it was semi-apparent from the title, but then when I started reading, it was obvious that it was the same people. So the paper in question is titled Chronic Stress Induces Alzheimer's Disease-like Pathologies through DNA damage, CHK1, Sp26 signaling. And this is a research group based in Wuhan, China, which obviously became very famous during the pandemic. So in the introduction, what the authors tell us is that there's been many epidemiological studies linking chronic stress to Alzheimer's disease risk. People who are stressed throughout their lives are at a higher risk of developing Alzheimer's disease later. And the connection, so it's very easy usually to talk about stress. Right? Look, I'm stressed and psychological stress and this and that, but what does that mean biologically? And for many years now, we've known that what is stressful to the brain connects to the rest of the organism through what is called the hypothalamus pituitary adrenal or HPA axis. The idea is the brain may psychologically detect a series of stressors that will wake up the pituitary gland, that will release a series of hormones, and some of those hormones, in particular ACTH, the adrenocorticotropic hormone, will go to the adrenal gland in the kidneys and induce the release of a whole bunch of what we normally refer to as stress hormones. Nowadays, cortisol has become really, really popular. Right? It's almost taking over dopamine, but it became like super famous as of late. But cortisol is just one of many glucocorticoid hormones that we collectively refer to as stress hormones. These glucocorticoid hormones target peripheral organs, and they explain many of the things that have to do with stress on various levels. So heart rate, respiratory rate, vasodilation, all of that in cases mostly of acute stress. So the famous fight or flight response, the accelerated heart rate, blood pressure, increased respiration rate, and so forth. So all of those things are induced by GC hormones released by the adrenal gland, but they're relatively short term, right? Now, that is not to say that those same GC hormones may have longer term impacts by acting on other organs, and in particular, one of the targets of these GC hormones is the immune system. So that leads to this idea that stress starts in the brain, goes through these stress hormones, which can then induce immune depression, and that is how typically stress can be associated with disease or with poorer outcomes, therapeutic outcomes during treatment, right? A person that is stressed may be more prone to getting sick, or they may be not responding to therapy as well because of these brain, adrenal, cortex, immune system axis. Okay? And what the authors stress in the introduction that is not that obvious is that in addition to targeting peripheral organs, these organs can go back to the brain and have effects on neurons themselves, right? Sort of, in a way, closing that loop. Stress starts in the brain, but then it can go back to it and have further effects. For instance, glucocorticoid hormones can induce tau hyperphosphorylation by activating GSK3 beta. And this model of chronic stress that the authors will use in the paper, chronic restrained stress, promotes inhibition of PP2A. And here I'm bringing back a very particular region of our network where both things we've touched on. So PP2 or PP2A can inhibit phosphorylation of tau, and GSK3 beta can induce phosphorylation of tau. And essentially what the authors are telling us is that stress can both, has been shown to inhibit PP2A, but it's the same, and it can induce, maybe I should keep the color coding, and it can induce GSK3 beta. So in both cases, it would be contributing indirectly to the phosphorylation of tau. I will introduce it a little better later, but this chronic restraint stress is essentially keeping animals physically restrained for long periods of time. And what makes this particular model stress is that it is chronic. Now, that is not to say that the animal is constantly physically restrained. The animal is restrained physically for several hours every day. And the way they do this is I haven't ever seen this, but apparently they place a rat inside of a plastic tube, and that way the rat cannot move, or a mouse, smaller tube, but that way these animals can't move. And they do that for several hours a day, and then they would get the animal out and then do it again the day after and the day after that, and so forth, right? All right, so the other angle of all of these that the authors introduce in the introduction is that there is precedence as well for connecting or correlations between DNA damage and depression, anxiety, chronic stress, and so forth, right? So what previous studies have shown is that there is more DNA damage in depression patients, and you can see more DNA damage in various models of animal models, that is, of chronic stress, right? So you stress animals through a number of means, and then when you look in their brain, you will see increased markers of DNA damage. And then the authors say, in a previous study we showed, and that's the paper that we discussed here during week 5, they showed that DNA damage accumulated in response to A-beta oligos and in brains of APP-PS1 mice. If you guys don't remember, these APP-PS1 mice are similar to those 5X FAD mice. These are mice that are prone to naturally developing Alzheimer's disease because they carry mutations that make them more likely to develop the symptoms. So the paper that we discussed in week 5 is sort of highlighted here. What that paper showed is that when they incubated neurons with A-beta peptides that will naturally form oligomers, that increased the activity or activated CHK1, which led to the upregulation of CYP2, and CYP2 is an active inhibitor of PP2A expression. So the idea was that DNA damage was stopping PP2, and stopping PP2 is what leads to tau hyperphosphorylation. And in that paper, we saw that they showed APP hyperphosphorylation as well, which led to the formation of more A-beta peptides. So if you need a refresher, and I don't think you do, but you can link back to week 5. So, in the intro, the authors end the intro by saying we know that stress is associated with DNA damage, and then we also know from our previous work that DNA damage can lead to a series of AD symptoms and phenotypes. So we hypothesize that stress is causing DNA damage, and that DNA damage is the intermediate that leads to tau and APP hyperphosphorylation, processing of APP into the toxic forms, formation of NFTs, neuron toxicity, and therefore AD symptoms. Okay? All good? Okay. So in order to start testing this hypothesis, they're going to go for the first part, if you will, of their model, right, of their working hypothesis, can stress cause DNA damage? They told us in the introduction that this has been observed before. So this is, again, one of those cases in which the authors are not necessarily trying to discover anything new. They're just trying to set up an experimental system that validates those previous observations. Right? So they're trying to see if in their hands, like we would say in jargon, so in their hands, they can cause stress to animals and see increased DNA damage. And for this, they use two approaches. One, there's an acronym that sounds funny, I'll recognize it, but it stands for Chronic Unpredictable Mild Stress Model or Paradigm, right? And we actually, I don't think I have time to show you, but coincidentally in my lab we study stress in flies, and one of the paradigms that we use is CUMS. So it's chronic unpredictable mild stress. And the idea is, as explained, I copied this verbatim from the paper. There is a combination of 0 to 3 kinds of stressors that were randomly applied each day. And the same stressor, there are some rules. You cannot use the same stressor in 2 consecutive days, and the total duration of this thing is 28 days. The idea here is there's a long list of stressors, all of which are applied in such manner that they cause mild stress to the animal. So the electric shock, it's only 2 seconds. The cold water bath is only 20 minutes. Water deprivation is only 12 hours. Each of these will be a mild stressor to the animal, will theoretically cause some mild discomfort, right? The basis of the CUMS model is that if you cause mild discomfort every day, and in many cases several times a day, because sometimes these authors apply more than one kind of stressor to the animal on the same day, over the long term, over 28 days, those animals are going to feel kind of miserable, right? Because every day they're suffering something that is mild, but it's every day. So that's the idea behind these CUMS models. And we do that with flies as well, although the stressors that we subject them to are quite different from this. There are some that are kind of funny, like moist bedding is very stressful to these rats. They don't like moist bedding, and I imagine it might be uncomfortable. Cage tilting, so that makes them a little nervous. Horizontal vibration, crowd living, that's a stressor that's pretty universal. If you crowd rats, or flies, or worms, in all cases, there's going to be a little bit of stress in the animal. So they use the CUMS model, and then they use this other one that I mentioned before, the chronic restraint stress, and what that is 3 hours of physical restraint a day over 21 days. Right? So with these two models, they're trying to test this idea that stress causes DNA damage, and ultimately the AD phenotypes. My question to you guys is, how would you check if stress causes DNA damage? What can we do to test that? Markers on the histones. Okay, so I'm going to say histone markers. Okay, so you said block the stress signals and then see if that stops the damage, right? That's yeah, I suppose that you could. Maybe I misphrased the question here. The question is about how can we determine that stress is indeed causing DNA damage? In other words, how can we see DNA damage in the cells? And then see if you block the stress. Now, remember that stress is your starting point, right? So blocking the stress is almost like not applying it. So it takes you back to the control condition, right? I guess in other words, what we're saying is, we want to see if stress causes DNA damage. How do you do that experimentally? Well, you take an animal that is not stressed, and then you look at whether the DNA is damaged, and then you take another animal that is stressed, and then you look at DNA damage. If you now say, now I'm going to block stress, that takes you back to the control condition, right? So it gets a little tricky that way. Now, I guess, and that's why I'm saying, apologize, I may have misphrased the question. So how do we look at DNA damage? How can we determine experimentally, yeah, I stress these animals and I see their DNA is damaged. Josh proposed looking at histone markers. How else might you look at DNA damage? A what? Comet assay. Do you care to describe it kind of briefly?
If it's damaged, it'll go faster in the.
That's really cool. That's right. Yeah. So, and this was the approach that was taken often before these histone markers, right? And the idea behind the Comet assay is if DNA is being damaged, it's being sort of broken into pieces. When you run that DNA through a gel, there will be smaller fragments that start running faster and deeper into the gel. So the idea was that intact DNA from an animal would all be compact and at the beginning of the gel, because imagine each of those chromosomes is gigantic, so they cannot migrate through the gel. But then if you had some DNA that was being broken inside of a system, then you would see these tails of smaller fragments that were being produced by damage to the DNA, which led to the formation of these images that to people doing these things look like comets, and we call it the Comet assay. Happy? Nucleases, that's right. But so what you're saying is, let's look at nucleases. If DNA is being damaged, there should be more nucleases around, so let's look at the levels of nucleases. Okay, so we'll get to that, right? The idea is we know that the steroids mediate the effect of the psychological stress that may be caused by having a moist bedding or having your cage tilted or something like that, right? So we know that those things that are very sort of psychological lead to steroids. I suppose you could say, well, I'm not going to tilt the cage, I'm not going to moist the bedding of these animals, I'll simply inject them with stress hormones. You could do that. These people decided to go let's go through all of the steps in order, right? So let's first start with psychological stressors, and as you'll see, they do go for hormones later. But I see what you're saying. What you're saying is let's bypass the stress itself, and let's just inject stress hormones. Yeah, they could have done that, but they went for the psychological stress. So, they ended up going for the histone markers. I really wanted to explore other possibilities because there exist other possibilities. And Gabby, just to tie up your idea of looking at nucleases, that's a good idea if you somehow predict that the DNA damage will be DNA degradation, right? So nucleases going and cutting the DNA. Not a bad idea at all. In fact, that happens. But let's remember that there is also other forms of DNA damage that are related to oxidative stress, free radical species inside of cells, which can cause chemical damage to DNA, which will in turn lead to mutations, chromosomal aberrations, and so forth. Lita?
I just had a question. So how would DNA damage marker work differently from a histone marker, or is this the same thing?
When we say DNA damage marker, we're typically looking at histone markers. So I personally don't know of any molecules or reagents that would allow you to distinguish between damaged DNA and healthy DNA, but cells do have ways to detect damaged DNA, and they send proteins there, and those are the things that we look for. And in particular, Josh was probably remembering a very famous mark for DNA damage, that is histone 2A. When DNA is damaged, the histone 2A is phosphorylated at a very specific position, and that tells the rest of the cell, hey, I see damage here, so you should come and fix this. And that's a popular way of looking at DNA damage, among other things, because it doesn't really depend on DNA being physically broken down, sort of cleaved. and because it also allows you for spatial resolution. So now the COMET assay, one of the issues with the COMET assay is that it happens in a gel, so you're not really seeing what's happening inside of the cells. Whereas with these histone marks, you can look through microscopy, you can look inside of cells. Okay, so they went for the famous H2A phosphorylation mark, and what they did is they stressed the animals either using the CUMS paradigm or the chronic restraint paradigm. They dissected their brains, extracted proteins from those brains, and they looked at the levels of phosphorylated H2AX. This is we call it gamma because it's a gamma phosphorylation. That's technical detail, but gamma H2AX is a very popular mark of DNA damage in mammals. And here are 3 different technical replicates, 3 different technical samples of controls and stressed animals through CUMS or CRS, and in both cases, there is an increase in the levels of gamma H2AX. I see some of your eyes squinting if you don't believe your eyes, because sometimes it's kind of hard to see these things in gel. Sometimes it's really so clear, but sometimes you need densitometry. Those are the densitometries of those gels in different biological or experimental replicates, so different sets of animals that this was done to. And in both cases, both CUMS and CRS were significantly or led to a significant increase in the amount of phosphorylated H2AV. Now, this, of course, was done in whole brains, right? So all that tells you is there is more DNA damage in the brain. But then the question becomes, where in the brain is there more damage? Is it everywhere? Is it in specific regions? Is it in specific cell types? So they took it to microscopy. In brain sections of either control animals or, in this case, animals subjected to this chronic unpredictable mild stressor paradigm, they looked at DNA damage, that's the green staining. They looked at nu-N, that's a marker for neurons. All neurons will express this type of this protein called nu-N, and you can use it as a universal neuronal marker. DAPI stands for, it's a mouthful, but it's a molecule that stains DNA. So it shows you where the nuclei of cells are. And then the emerge image is combining all 3. So I will go first with this image from the hippocampus. I know that it may be a little kind of hard to appreciate, but when we look at the merge image in the controls, you see a lot of reddish, pinkish cells in the cells of the hippocampus, whereas the same cells in the stressed animals all look a little more of that lighter blue, and that lighter blue is presumably leaning towards whitish. That is presumably because we have a more superposition of green and red and blue, all colors. Whereas here, there is less of that green, less of the DNA damage, therefore you have that purplish color that comes from only the red and the blue. Okay? The same happens in a different region of the hippocampus, the dentate gyrus. And once again, if you look at these bands of cells, there is very little damage in the control animals. I should use the pointer here. Very low damage in the control animals and a lot more damage in the stressed animals. You can take these images and subject them to computer quantification, right, of the different types of fluorescence. And in both cases, caused a significant increase in the relative intensity of that DNA damage mark. Based on these images, the authors also claim that the DNA damage is happening primarily in neurons. Why do you think they're making that claim? Based on these images, where do you think Sure, but the hippocampus and the rest of the brain will have also glia, right? So where do you think this claim that DNA damage is happening primarily in neurons is coming from? Exactly. So the idea here is that it was that increase in the overlap between the green and the red that leads them to say that there was increased damage in neurons. But the authors take it a step further. They say that it happens primarily in neurons. So what type of evidence would you like to see to back that claim up? What would convince you that, yeah, damage is happening primarily in neurons? What experiment, if you were a reviewer of the paper, and the authors show you that there is more damage in the neurons because there is increased overlap between green and nu-N staining, which I'll take. But then they take it a step further and say damage happens primarily in neurons. What would you say? Oh, hold on a second. You should show me. What would you like to see to support that claim? Excellent. And that's something that they did not share with us. So in my humble opinion, that was a shortcoming of at least this part of the paper, right? Had they shown us a marker for, let's say, microglia or astrocytes or oligodendrocytes, and we've seen these markers, they exist, we've seen them along the way, in the red, and in those cases there is very little overlap between the red and the green, then I would say, okay, cool, now I believe you. But they didn't do that. So, but they'll continue with this idea that DNA damage is primarily in neurons. Remember how I said they used two models of chronic stress in animals? There was that COMS model that we just saw, and then there was the CRS, the chronic restraint stress model. For some reason that, again, the authors in the paper don't explain very well, and I'm not quite sure why. For this particular experiment, the staining for DNA damage in brains of animals that were subjected to stress, instead of trying the CRS, the chronic restraint stress paradigm, they tried ARS, and ARS stands for acute restraint stress. So all it is just having the rats one day, and not even 3 hours, 2 hours inside of those tubes so that those rats couldn't move, and then a day later, I think it is, dissect their brains and look for damage. Okay? And what they see is that even after one acute stressor, there is this significant increase in the relative intensity of H2AX that happens, once again, in the CA1 region of the hippocampus. So in controls, there is no staining whatsoever, nothing to see here. But then in the acute stress animals, there is now all of a sudden a lot of signal coming from that region. And this thing here, the emerge, goes from essentially pinkish, purplish to whitish, indicating that there was a lot of DNA accumulation. The authors don't address this in the paper, but I thought it was interesting to compare the extent of DNA damage increase that they saw with the acute restraint stressor versus the chronic unpredictable mild stressor that takes over 28 days. And as you can probably appreciate, I kept the scales the same as they are in the paper, right? So I didn't modify anything. What do you think? Is the damage caused by 1 day of stress and damage caused by 28 days of stress the same or different? Or, yeah, right? We can't say for sure, right? But intuitively, it's 130 and 145 maybe percent increase, or 145% of the relative amount. Who knows what, had they done this really back to back, because they probably didn't, right? But had they done this really back to back and compared them statistically, our intuition is that probably they would not have been different. So the question becomes, is it chronic stress or any stress that is causing DNA damage? And it's an important question that again, it won't come back up in the paper, but it's related to this more general question of how do we define chronic stress. And I thought this was a good place to have this conversation, because what I'm about to say about stress really applies to any other experimental manipulation or environmental condition a system, an organism, a cell, a tissue can find itself in. And that is the difference between chronic and repeated. Because how would you guys define chronic stress? If I asked you to define chronic stress. So, but let me take you back to this idea of long term. So something can be long term and discontinuous, or long term and continuous, right? So for instance, I think many of us can relate with the idea that school or work or both it's long-term stress. But when we're not at work, when we're not at school, we've discontinued that stressor, if that makes sense, right? So it's kind of hard sometimes, yeah. Well, so continuous exposure versus repeated exposure to the stressors, both of them can be chronic. And what I'm saying now, it's a discussion that exists in this field, and the reason I'm somewhat familiar with this discussion is because we've had to sort of struggle with that conversation ourselves when thinking about chronic stress in flies, right? There is a difference between saying, for instance, let me give an example. There is hypoxia. Hypoxia is reduced levels of oxygen, right? You can have an organism under chronic hypoxia. And what we mean by that is you place that organism or cell or tissue or sample under less than 5% oxygen tension, and you keep it under those conditions for a really long time. That's chronic hypoxia, no question about that. But then you take a stressor like sleep deprivation or aggression, or how do you make aggression chronic, truly chronic? That's hard to picture, right? Or starvation. You can go past 2 days and you basically kill the animal, right? So how do you make starvation chronic? Well, one of the ways that you make it chronic is by actually having a stressor that may be discontinuous by repeated, right? So sleep deprivation may not necessarily be chronic, but if there is an individual who every night gets only 3 hours of sleep, that's chronic sleep deprivation. Right? Even though we know that they will sleep 3 hours a night, as a stressor, it's chronic even though it is discontinued. So it's repeated as opposed to nonstop. Okay? So the reason I bring this up is because seeing that one bout of stress caused the same level of damage opens up the question of, was it what they saw with COMS, was it a consequence of really that repeated chronic nature of the COMS paradigm? Or is this simply reflecting the very last stressor they saw yesterday? Right? And there are experimental ways to tease these apart, but the authors either did not do those experiments, they were sort of more inclined to pursue different routes. But from a behavioral biology point of view, there are some things that you can do to see if COMS is bringing something else other than the damage that just using one stressor one day would cause. Because otherwise I could say, well, what I see with the COMS animals is really a reflection of what they experienced last day, or the first day for that matter, right? And that the chronicity in the stress is not really adding anything. I believe that 24 hours after. So just like the NARS, the ARS model, they sacrifice the animals the day after. In the COMS model, they sacrifice them on day 29th. Excellent. That's exactly, yes, that's exactly what I'm talking about. So you could have a modified COMS group in which you subject them to the first stressor, then wait 28 days, and then sacrifice them, right? Or see how long after one or two stressors they can go before they recover. None of that was done. None of that was done. And then there are other things that weren't done related to this, and that is ARS. This is the only experiment that they used it with. All of the subsequent experiments that we'll discuss next, in all of them they used CRS, not ARS. So we go back to the idea of I'm looking at chronic stressors that we know could be, we know that in neither case they're permanent because the animals are constantly going through periods of rest. So in chronic restrained stress, it's not the animal is trapped for 21 days. No, they're trapped 3 hours a day for 21 days. There's a difference, right? So could these chronicity in these stress models bring something else that ARS would not have triggered? We won't know because they didn't do any more experiments with ARS. Okay. I'm not necessarily trash talking the paper or anything. I'm just pointing out to so often when you're doing experiments in a real experimental setting, at some point you need to make some decisions as to what you're not going to pursue, and this seems to be one of them. So they decided to use ARS for I don't know why, to look at the histology, and then from here on out is all chronic stress. Okay. So, Josh, in a way, going back to your comment before, can you just inject the stress hormones instead of stressing the animals? That's what they did, but instead of the whole animal, they did it with the neurons in culture. Because now they wanted to get into the biochemistry of how stress affects neurons. So how do you stress neurons? Right? A whole animal, you can tilt the cage, you can wet the bed, but how do you do with a neuron? What stresses a neuron? So what they did is they took the neurons in culture and they incubated them with a few, but they tried with two stress hormones, corticosterone and corticotropin releasing factor, both of which are known to be one of these systemic stress hormones that will go and impact peripheral tissues and target organs and so forth. And they tried with both. Corticosterone was a little too toxic to the neurons. It was killing the neurons in culture, so they stuck with CRF. So when they took primary neural cultures from animals and they incubated them with CRF, they saw a bunch of things. First, they saw that they could induce DNA damage, CHK1 activation, and CIP2A upregulation. All of that is shown in the left panels here. So there was increased amounts of phosphorylated H2AX. There was increased expression. I know that you may have to squint your eyes, but increased, not expression, but levels of the phosphorylated CHK1 proteins. And as a reminder, when DNA gets damaged, CHK1 gets phosphorylated at these two positions, serine 317 and serine 345, which will induce some sort of conformational activation that will activate CHK1 itself. Here is a control for CHK1 levels, and we've seen this before. When you're looking at higher levels of phosphorylated CHK1, how do you know that it is the phosphorylated form and not CHK1 overall? So what you do is you run a Western for CHK1, the protein, and you see that those levels remain constant, but the phosphorylated forms are elevated. So that's how you can make a claim that phosphorylation of this protein is increasing. And then this one is probably kind of rather apparent. the increase in the expression of Sp2A. And Sp2A was that protein that inhibited the PP2 phosphatase, acting down here as a loading control or housekeeping gene across the samples, right? So with this, they see that there is an increase in DNA damage and the subsequent expected activation of CHK1 and upregulation of CYP2, right? With the same animals, they looked at tau phosphorylation and APP hyperphosphorylation. So for when it comes to tau, those 4 panels show, again, hard to see by naked eye, but presumably an increase in the phosphorylation of tau at those positions, threonine 231, serines 396, and 404. And then tau 5 is simply looking at tau, the protein, as a whole, so to make sure that the increase in the phosphorylated forms wasn't an overall increase in everything, but only the phosphorylated forms. And with APP, they looked at that famous, I don't know if you guys remember, but that famous phosphorylation at threonine 668 that makes APP more readily cleavable by gamma secretase, which will generate the toxic forms of A beta peptides, right? So when APP gets phosphorylated at that position, it's going to be processed primarily to the toxic forms. An APP blot down here to show no significant changes in total levels and acting as a housekeeping loading control as usual, okay? Now, if you don't believe everything that I'm telling you based on these images, which I gotta confess, I have a hard time seeing any of these by looking at these plots. Many of them are a little too overexposed for the human eye. They do offer us the densitometric analysis for all of these things. And in all cases, we're not going to go through details, but in all cases, the blue bars that correspond to cells treated with CRF are significantly higher than the red bars that are the controls. Okay? So, incubation of neurons with CRF, which is almost like telling neurons, you are in a stressed animal. Increases the DNA damage, activation of CHK1 pathway, and phosphorylation of tau and APP. And they also saw that these neurons now release more toxic A beta 42. So they looked in the supernatant of these cells and they saw an increase in the A beta 42 present in the supernatant. And even though the difference was not statistically significant, they saw a concurrent decrease in the amount of A beta 42 they could see in the cell lysates, meaning how much was left in the cells. All right, so a few slides ago, we sort of showed the stress connected to DNA damage through Western blotting and microscopy. Now we see that we bypass the psychological stress, but we go for CRF stress, if you will. And we can link that to both DNA damage and the APP and tau hyperphosphorylation. Putting it all together, the model becomes what, in a way, they hypothesized at the very beginning, that stress causes DNA damage, and it is that DNA damage that causes the AD phenotypes. How do we test that model? Exactly. Take out the intermediate, take out the miniman, right? So you said stress the animals or CRF, right? If you're going to be looking at all of these things that are very cellular, biochemical, which one would you do? Animal or CRF? CRF, right? You stay in culture so that you can look at the biochemistry of all of these. That's exactly what they did. They incubated the cells with CRS, shown here, but they also had some cultures where they had CRS. Wow, I think I take it back. So this is CRS. They did it with animal brains. I forgot. So this is not CRF in culture. This is actual CRS. So this is chronic restrained stress animals. And then to some animals, they gave this drug called GDC, that is a CHK1 inhibitor. And to some of the other animals, they gave them vitamin C. And vitamin C is, if I ask you, what does vitamin C do? What immediately comes to mind? Scurvy, yes, yeah. A little less extreme than scurvy, a little more 21st century. Why do we take vitamin C, or when do we take vitamin C? When we're sick, right? Yeah. We associate it with something, that is good for the immune system, will keep the colds away and whatnot, right? It turns out that vitamin C is a fairly potent antioxidant. Okay? So the reason for using vitamin C as a protector of DNA or protect against DNA damage is that what damages DNA is often these reactive oxygen species, right? These ROSs that are somehow, that's a huge black box there, but somehow elevated by stress and the stress hormones, right? There's going to be, I'm sure, something with the mitochondria and metabolism and this and that. But the point is that what damages DNA, what damages proteins is typically oxidative species. And vitamin C is a general antioxidant, so they figure, well, if we have the animals under stress, but we give them a lot of vitamin C, we may be protecting their DNA, right? And the same with GDC. The idea was if they're thinking, well, DNA damage will activate CHK1, they have shown it too, right? But if the hypothesis is DNA damage activates CHK1, and it is CHK1 that leads to APP phosphorylation and tau via the upregulation of Sp2A and downregulation of PP2A, then if you take out that middleman, CHK1, you should see those effects go away. In the control, the control is rats that you don't do anything to. You don't stress them, and you don't give them CHK1 inhibitor, you don't give them vitamin C, just rats. CRS, rats they kept 3 hours a day for 21 days inside of a tube, and CRS plus GDC, CRS plus VC or vitamin C, same animals except that in the drinking water you kept giving them either the drug or vitamin C. So the idea here is, as Gabby was proposing, you cause the stress, but then somehow you inhibit DNA damage. They took two approaches to this, and then you ask the question, what happens to the downstream effects? And just to make sure that DNA damage was as expected being inhibited, they looked at the marker for DNA damage, and they saw that it is reduced with both the CHK1 inhibitor and vitamin C. The markers for CHK1 activation, meaning when CHK1 responds to DNA damage, was also reduced under both conditions, elevated by stress, but then negated by both drugs. And upregulation of Sp2A, very apparent in stressed animals, and it's gone in animals that are stressed, but that are given vitamin C or the CHK1 inhibitor, right? Yes. Why? I don't know. We didn't know back then. I still don't know. and it'll come back. Last slide is for you. Last slide is for you. Yeah. I didn't want to point anything out, but you notice, yes, again, this business of CHK1 activation is supposed to be downstream of DNA damage. So if you inhibit CHK1, the enzyme, you shouldn't be altering any of the upstream steps, right? Any of the causes that led to CHK1 activation. You can inhibit CHK1, the enzyme itself, and anything that happens after. But how would you be inhibiting? That's totally putting the cart in front of the horse, right? So we'll go back to that. For now, bear with me. They're saying that using these drugs inhibits DNA damage. How? They don't explain. And then what they're really interested in is will all of the other things that we saw, APP phosphorylation and tau phosphorylation, go away? And once again, in this case, the blots are a little better, in my opinion. So tau phosphorylation is diminished, APP phosphorylation is diminished, and I'm not showing them now because it was a truckload of graphs, but they did the densitometries, and in all cases, the measurements backed up this idea that stress is causing DNA damage and the phosphorylation of APP and tau. But if you give an antioxidant or this CHK1 inhibitor, all of that goes away. So it seems like, indeed, if you take out that middleman of DNA damage, the rest goes away. And this experiment was important because, who knows, maybe stress is causing DNA damage. We can believe that part, but it may be causing something else that is truly connected to the AD-like symptoms, right? So by taking out DNA damage and seeing that the AD-like symptoms go away, totally reinforces this idea that it is DNA damage that is the intermediate between the two. Questions, anyone? How are we doing? Good? Okay. And I should have shown that right away, but I'll show it now. They also looked at a beta-42 release into, in this case, it's simply the amounts of either soluble or oligomerized, a non-soluble fraction of AB42. And in both cases, what they see is that there is a significant increase with stress, but both go down when the animals drank vitamin C or the CHK1 inhibitor. So all AD phenotypes were being inhibited by these things. So throughout the semester, we've seen that there is all of these things that researchers do to look at the biochemistry associated with AD, A-beta production, oligomerization, clearance, secretion, blah, 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 tau phosphorylation, NFT formation, blah, 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 blah. But then eventually, at some point, people want to know about neuronal function, about brain function, because that's ultimately what AD affects, right? So they decided to test two things that are associated with neuronal function. One is anxiety and depression. And the reason they chose anxiety and depression is because anxiety and depression has been associated so many times with chronic stress. Right? That's one of the things that chronic stress will cause or manifests itself. That individuals, rats, humans, possibly flies, become anxious and depressed. And for that, they chose 3 behavioral tests. One is called the open field test. And in the open field test, what you do is you basically place the animals in a sort of a round arena, and you record them from the top, and you see how much time they spend exploring the center of the arena versus how much time they spend on the periphery of this thing. And an animal that is anxious and depressed will move mostly in the periphery. They won't explore the center because the center is scary. And for an animal that has been under constant stress for weeks on end now, they're just too scared to venture into the center, right? So even though there's 2 graphs here, the first one shows the total time that they moved. So this history of stress did not affect their ability to move. These animals can move, but they choose to spend significantly less time in the central area than controls, right? So stressed animals spend less time in the middle than controls, and that is because they're scared, they're anxious. Treating the animals with either drug, vitamin C or the GDC drug against CHK1, did not change that. So GDC had no effect on anxiety and depression, and vitamin C had no effect on anxiety and depression, at least as measured by these particular behavioral tests. That isn't the only behavioral test they used. They also used what is called a tail suspension test. I had to look this up. So you take the animal and you tape them by the tail to a rod. So now they're hanging from their tail, and for 3 minutes, you basically count the number of seconds that they're struggling and trying to get themselves out of that situation versus just hanging in there, right? So the more time an animal spends hanging, not really doing anything, in other words, immobile, that's an indication of depression. I know. And these things are presumably conducted with all the ethical check marks and so forth. But it sounds like a little extreme, but that's what the test is. The free swimming test, similar. You basically dump the animals in a tank with some liquid, water, say, and you measure the amount of time that they spend swimming. And a depressed animal won't even try. They won't swim much. They won't sink and drown, but they spend a lot less time trying to get themselves out of that situation. Right? So again, immobility time. So in both cases, there was a significant increase in immobility with stress. So these animals are depressed. And here's where the water split a little. Vitamin C actually, when it comes to the tail suspension test, induced a partially significant drop in immobility. So it partially protected against depression as measured by these behavioral tests. The p-value is 0.07, so it's just a tiny bit over that famous threshold of 0.05. And then with the free swimming test, same thing. You can see a drop. It's not statistically significant, but it's kind of close. The argument often in these cases is, well, who knows? If we had a larger sample size, we might pass significance. So what the authors say is that inhibiting CHK1 certainly had no effect when you look at the comparisons between GDCA1 and the stressed animal alone, no difference. But then vitamin C, they say, may have some partial protection, some partial effects. Okay? Anxiety and depression are things that we typically associate with stress. And seeing that the stressed animals are anxious or depressed is a good sign that the stress paradigm is working. But anxiety and depression is not necessarily what we associate with Alzheimer's disease. What is it that we normally associate with Alzheimer's? Anyone who hasn't spoken yet, but get this one. Sleep, okay. All right, sleep would have been a good one. We know that Alzheimer's relates to loss of sleep and circadian rhythmicity. Good. They didn't do it, but that would have been a good idea. What else? When you think of an Alzheimer's patient, what is it that immediately comes to mind? Memory, right? Memory. That's the sort of flagship neurocognitive defect associated with Alzheimer's. So they did a memory test. And this test is kind of fun. So the animal spends many days, let's go to blue, many days in a cage where there are two objects. Let's say I suppose a cube that we call A and a sphere, a ball that we call B. And the animal is trained for several days in that cage with those two objects. So on day one of the actual test, you will place some of the animals in a cage that has the usual, the A and B, the cube and the ball. And then you'll measure how much time the animal is inspecting these two objects, right? And because you train them under those conditions, control animals will spend the same amount of time inspecting both. That's exactly what you expect from a regular rat, that it will be inspecting both the same amount of time because it's used to both. Okay? But then you take those control animals and you place them in a cage where there is the usual cube, but now a pyramid. Right? So a control animal will see that as a novelty. And rats are curious, like cats, like human beings, like all other animals. Right? They're curious. They want to see what that new object is about. They need to figure out if it's a threat, if it's something that they could eat, and so forth. Right? So you will see that among control animals, the animals will spend a lot more time inspecting that novel object. So that's why this is a novel object recognition test. So the idea here is that in this left panel, all you're doing is making sure that the animal got used to both objects. And in the right panel, what you're doing is you're sort of getting a sense for how much they appreciate that there is a new object in their environment. When the animal is spending a lot more time in the new object, that means the animal is healthy and it's recognizing that as a new object, and they'll go and spend a lot more time inspecting it. But notice what happens to the stressed animals. The stressed animals show no difference, no significant difference in the time that they spend inspecting both. They didn't even notice that there was a new object. And that, in the context of AD, is a reflection of memory loss. Right? When you stress the animals, but you give them the vitamin C or the CHK1 inhibitor, now you've rescued or you're restored the ability of the animals to recognize novel objects, meaning that they kept their memory, right? So from this, the authors conclude that both GDC and vitamin C protect from memory loss. All right, we have a couple of minutes, but I wanted to go through these real quick. So in the discussion, what the authors argue is that stress is causing DNA damage. And because there were some differences in the behavioral tests, what they claim is that the DNA damage is, on the one hand, triggering a CHK1, CYP2A, PP2A pathway that leads to the accumulation of APP phosphorylation, tau phosphorylation, A beta secretion, etc., that leads to cognitive defects, right? And then separately, there is another pathway through something else that they haven't identified that leads to anxiety and depression. And the idea here is that vitamin C is protecting against DNA damage, so it will somewhat protect against both, though the authors had to spend some time explaining why some anxiety and depression tests, vitamin C didn't show an effect, but for some others it did. So they argue that there may have been something about dosing and so forth. But basically what they're saying is vitamin C can protect against both. So vitamin C might be getting to the root of the problem, and it's protecting against DNA damage. Whereas GDC acts only on this pathway. And that's why it protects against cognitive defects, but it cannot do anything against anxiety and depression. That's what they discuss. But now I'd like to bring back what Asosi was saying a few minutes ago, that GDC was also protecting against DNA damage. When the mice were treated with GDC, with this CHK1 inhibitor, DNA damage was gone, at least as revealed by the H2X phosphorylation. So what that means is that both GDC and VC must be protecting against DNA damage. How do you explain then the differences in effects? So what I think is that what we may be missing here is the stress is splitting up into two different pathways earlier than DNA damage, right? And that vitamin C is protecting against DNA damage, fine, I can buy that argument, but it's also protecting against that something else that is going on. Whereas GDC is only protecting against DNA damage. Okay, so this model would be a little more consistent, not so much with the behavioral data. The behavioral data would support either model. But this model would accommodate the fact that both GDC and vitamin C protected against DNA damage. So we go back to how could vitamin C protect against something else? And based on what I told you about vitamin C, what do you think? Or based on what you told me about vitamin C? What could vitamin C be doing in the animal that is not related to protecting against DNA damage itself? Yeah. So who knows? Jacob, you were going to say the same thing? Right? Yeah, we know that vitamin C is an antioxidant, and sure, ROSs can cause DNA damage, but they can also cause mitochondrial damage, and they can cause protein damage, and they can cause who knows what else damage. Changes to metabolism. metabolic changes. So the idea here is maybe vitamin C is a broader protector against the damage from stress, and it can, they partially correct both the cognitive defects and the anxiety depression.